Okay. Good afternoon, everyone. Can you hear me? Yes, if I can have your attention. So welcome, everyone, dear colleagues, ladies and gentlemen. It's a great pleasure to see you here for another Mendel lecture within the 2018-2019 series. And it's my personally pleasure to introduce today's speaker, Professor Patrick Sang. So I'll also share a few introductory words, as is typical for us. Is it on, right? Okay. So Patrick received his Bachelor of Science degree at the University of Liverpool, then followed with the doctoral degree at the Oxford University, both in the field of biochemistry. And then he completed his 8-year postdoctoral stay or training fellowship at the University of Rochester in New York with Louis and Satya Prakash, where he really played an essential role in characterization of some of the key components of nucleotide excision repair. So that's a DNA repair pathway required for the repair of bulky lesions and cross-linked strands of DNA, but also characterizing the role of some of these similar proteins in RNA polymerase II transcription as well. Since 1993, he received his independent position at the University of Texas in Galveston, and what always impresses me is that within the year of his independent position, he published a single author science paper describing the role of one of the key factors of homologous recombination in strand exchange. I don't think I've seen many of single author Nature or Science paper. So this is really an achievement. And then since then, in I think it was 1997, he joined the University of Texas Health Science Center in San Antonio as an associate professor, and then was appointed as a Zachary Distinguished Professor at the Molecular Medicine. And he also co-directed National Cancer Institute funded training program for 2 years or 3 years, and then since 2003 he joined Yale University Department of Molecular Biophysics and Biochemistry in Connecticut, where he also served as a chair for 2 3-year terms. The same department, including many of the members of National Academy of Science and also Nobel laureate. And then the kind of closed the circle. He actually recently, since September, joined back or returning back to Texas, University of Texas in San Antonio, where he has appointed several positions, and I have to read it out just to make sure. So he will occupy Robert Welch Distinguished Chair in Chemistry. He'll be appointed Professor of Department of Biochemistry and Structural Biology. He will serve as Associate Dean for Research at the Joe and Teresa Lozano Long School of Medicine, and he will lead a new research program in genetic integrity at the Mays Cancer Research. So very impressive list of positions he's occupying at University of Texas. So during his career, independent career, I already mentioned that he, from the postdoctoral stay focusing on nucleotide excision repair, he moved into homologous recombination, and that's going to be topic of his talk. He really played on his own, and of course people in his laboratory, essential role in characterizing critical steps and components of the mechanism of homologous recombination. As I mentioned, the RAD51 recombinase, which is actually searching for the homology, its conservation from yeast to humans, and also characterized the fundamental role of tumor suppressor proteins, including BRCA1, BARD1, BRCA2, PALB2, Bloom helicase, and also components of Fanconi pathway, and really provided essential characterization in characterizing the key role of homologous recombination in cancer avoidance. And as maybe one of his colleagues pointed out, that his approach is disassembling the components, isolating them individually and putting them back together. So I'm really looking forward to disassembling the engine here within the lecture, and that we will understand the mechanism a bit better. But before I do so, I would also like to congratulate Patrick to his outstanding achievements in science, and would like to present you with a Mendel Medal. So Congratulations.
I've been teaching Mendel's law of heredity and so on for quite a number of years. But I have to tell you, I mean, looking at the original manuscript, I'm nervous here, okay? If I stumble, you will know why. Because I'm nervous, I'm overwhelmed. I mean, this is really the original house of genetics here. And I'm just overwhelmed. Thank you so much for inviting me. I don't know what I've done to deserve this, but I'm here, so I'm enjoying myself. Okay. We work on a lot of stuff. I'm not going to try to cram everything in my presentation. I'm going to cherry pick a few stories, all right, to focus the health relevance of what we're doing more. I'm going to do a lot of introductions because I know that there are a number of undergraduate students in the audience and also a large number of graduate students as well. So I'm going to do a lot of introduction, giving you the background and so on, before I swing to talking about hard data and so on and so forth. For the past 25 years or so, we've been focusing on this particular problem. Until very recently, we were a yeast lab, using the budding yeast as the model system. And it has been very beneficial in terms of understanding some of the intricate steps in homologous recombination and the engagement of homologous recombination as a DNA repair tool. However, many of the components found in mammalian cells, they're simply not present in yeast. I mean, they're completely absent. You cannot even remotely think of an awful lot of these proteins. I'm going to talk about some of them, including BRCA1 and BRCA2. There's just no clear awful lot, okay, in lower eukaryotes, which really promise to go to the mammalian system, okay, to try to figure out how these tumor suppressors contribute to the DNA repair process. I'm going to give you a lot of history and background and so on, giving credit where it's due, okay, before I move on to talk about our own study. All right. We study DNA repair from the standpoint of understanding the relevance in cancer biology. We have to. When we write grants, we send a grant to the National Cancer Institute, the first thing they ask you is that why should we give you whatever, $1 million or $2 million or what have you to do what you want to do? Okay, so this would be our mission statement. We want to understand the role of chromosome damage repair, okay, in cancer biology, more importantly, in tumor suppression. I just want to point out, many of you probably know this already. Cancer remains a leading cause of death. Okay, for instance, in 2009, these are real data from the World Health Organization. 1,500 people a day die of cancer in the United States alone, all right? If you look at the pie chart, unfortunately, cardiovascular disease remains the top killer. I think McDonald's and so on have something to do with this. I bet you, 100 years, 150 years ago, if you had the wherewithal to compile a pie chart like this, injury, okay, right here probably is would have been a major cause, okay, because of the nature of employment and so on and so forth, and then chronic disease and so on. This might have been half, okay, of the pie chart and so on. Cancer might have been, even more prominent, all right? But nonetheless, cancer remains a leading cause of death, okay? It's only getting worse as we're getting older, so on, because part of the cancer issue is really related to aging as well. If you live long enough, right, the chance of you getting cancer is, significantly higher. Young people, you don't need to worry. We have to start worrying about that. Okay, all right. So I'm going to discuss cancer biology from the standpoint of mutations and chromosomal rearrangements and so on. I'm not going to show you any cytogenetic data and so on, but it's important to understand that this type of large-scale rearrangements of your chromosome is a major cause, okay, of cancer. It's not disputable anymore. There's just overwhelming evidence that that's the case, all right? Now, DNA mutations and chromosome rearrangements mostly stems from a failure to repair DNA damage or failure to repair DNA damage properly, the use of a wrong pathway to repair something, that sort of thing. Now we know that problems encountered during DNA replication is a major cause, okay, of DNA mutation, slip-A mutations, insertion mutations, what have you, point mutations, and also under some circumstances, it can give rise to chromosomal rearrangements when replication fork collapse, okay, when they encounter a certain type of lesion. Now, I don't mean to suggest that DNA mutations and so on are the only cause of cancer, of course. Viral infection, bacterial infection, and so on represent, a major cause of cancer as well. It's debatable, okay, until recently. I think there's so much evidence now bacterial infection can also directly lead to cell transformation in cancer. Now, changes in hormone levels and types and so on are also associated with certain types of malignancy. I will refrain from saying too much because I'm ignorant about these, okay? I know something about this particular, subject, so I'm going to just focus on that today. Even though I teach this stuff at Yale, I'm really quite ignorant about them, all right? But I need to project an image I know something because people pay a lot of money to send their kids to Yale. Okay. My confession. Try to keep it within this room. It's not possible because it's being streamed. All right, okay. Now, DNA damage stems from, a variety of sources. Okay, not to scare you, while we're sitting comfortably in this room, these things happen to our DNA. We're talking about the number of events per cell per day. There are 5 or 6 trillion cells in our body? You can do your math, all right? 50,000 single-strand break, okay? Depurination resulting in, all the bases and so on. You add them all up and times 5 or 6 trillion, what would be gazillion, okay? I don't even know, what number to use, all right? So we have to deal with a lot of DNA damages. Today I'm gonna focus on DNA double-stranded break. Rather rare, but they're very cytotoxic and genotoxic for the reasons I'm gonna lay out for you, all right? Is this working okay? All right, okay. Now, what am I talking about here? If DNA damage occurs so frequently, all right, and DNA damage has the potential of giving rise to, chromosomal rearrangements and cell transformation and so on, why don't we all get cancer? Why would we even be born to begin with? This is because we have DNA repair, all right? We have different types of DNA repair. I won't have the time, nor is it the purpose for me to dwell on all different types of DNA repair today. I'm gonna focus on one type of DNA repair, double-stranded break DNA repair, that we have been working on the past 25 years or so, and have been making contributions as we go along. All right. Now, where do these breaks come from? All right. DNA double-stranded breaks can be induced by high-energy radiation, chemicals that are prevalent in the environment, the food that you eat, the beer that you drink. It can be converted into aldehyde, which can damage DNA and indirectly lead to double-stranded break. You should enjoy your beer. Don't worry too much about that. The amount that we consume is probably okay, all right? And cellular metabolites, stemming from cellular metabolism and so on, you can avoid that. It's the cost of doing business, basically. You shouldn't even worry about that, right? But nonetheless, normal metabolism can generate reactive intermediates that damage your DNA. This is very well established now, all right? Now, more and more, we now realize that double-stranded breaks can also stem from failed DNA replication. So when DNA replication ensemble, the DNA polymerase ensemble, let's say, run into a single-strand break can stem from DNA repair reactions. For instance, if you alter base, the repair system will remove the base, generating a single-stranded break in a transient fashion. But if such so happen that DNA polymerase comes upon that break, you get a collapsed fork. Basically, it's DNA double-stranded break with a single end. A chromosomal break will have 2 ends, a collapsed fork will have 1 end. I'm gonna touch upon that a little bit more, later on. And also when DNA polymerase ensemble runs into a bulky adder, it stalls, and then the stalling leads to signaling events and so on. Some cellular nucleases can attack that particular structure and also generate a DNA double-stranded break under those circumstances. So breaks are coming from all these sources, especially when cells are proliferating actively. All right, so unrepair or misrepair double-stranded break, induce cell death. That may not be the worst consequence, but it can also cause chromosomal rearrangements, cell transformation, and cancer, also neurological disorder, but that's not my focus today. We really focus what we learn from the standpoint of understanding cancer biology. But if you talk to the neuro folks, they will tell you that DNA repair actually has a big deal. It's a big deal in terms of understanding the health of the neural system as well. All right, so what type of chromosomal aberrations or rearrangements can be induced, okay, by unrepair or misrepair DNA double-stranded break? The simplest being a gap, okay, you're just losing some information from the chromosome, or static breaks and so on. Sometimes a telomere can be added, okay, onto the end of the break in a de novo fashion and so on. And that can also lead to, okay, other types of chromosomal rearrangements as cells proliferate. You can get a translocation. This happens quite often, okay? This is a big deal in terms of understanding, okay, loss of heterozygosity in disease pathogenesis and so on. Now, at times, instead of, okay, repairing the break, okay, by joining the right piece of chromosome, the chromosome becomes, joined end to tail to give rise to a ring chromosome. You can also get a dicentric chromosome with an acentric fragment. All these are found in cancer cells. We know that, most of us, okay, are aware of this. Now, this particular slide lists several types of chromosomal rearrangements. They're even more exotic, okay, type of chromosomal rearrangements that stem from, okay, these initiation events, okay, that are often found in cancer cells. All right, enough introduction. I'm going to start talking about, okay, what we have been focusing on, but I want to give credit to people whom I admire, whom I think are really the heroes, or in this case, a heroine, okay, of our field. So Maryclaire King, 20-some years ago, she persisted, against a lot of criticism from contemporaries in the field. People thought that she wasn't really thinking right, a little bit crazy, people thought, okay, because she thought that breast cancer has a familial origin. She persisted with that idea and using very careful chemical linkage analysis and so on. Lo and behold, she was the person who identified the first genetic locus linked to familial breast cancer. Okay, she named the gene breast cancer 1 gene. So when she was interviewed by a HuffPost, a sort of like a weekly news outlet sort of thing, he said, yeah, it was a long road to walk. For instance, the week that my husband left me, all right, and my house was broken into. Finally, I got a grant to study, this particular genetic locus that ended up being BRCA1. Two things that become very obvious: she's brilliant. She didn't care about criticism and so on. She believed what he was doing. The other thing that's clear, okay, I want to point out, she clearly didn't need her husband, all right, to achieve what she achieved. Okay, that's a good lesson to learn, all right? Despite the fact that, or maybe because of the fact that he left, right? I don't actually know the details and so on, but I thought it was a little bit amusing. She's a really remarkable person. I don't know how much of, well, young people may not know this, older people know. There was a dirty war, sort of not very well advertised until very recently in Argentina. Basically, there was a lot of political unrest, and then the military people in charge of the government then basically just made people disappear. Okay, that was termed the dirty war. Like 15,000 people disappeared. Many of them were women. They gave birth, to babies and so on when they were in prison. Those babies disappeared also because they were given to police officers, military officers, and so on, who didn't have their own children, all right? So these abuelas, okay, Spanish word for grandmothers, they got together, okay? They wanted to launch this initiative to find their missing grandchildren, came along and used mitochondrial genetics to help locate like several hundred kids and reunited them with their families. So it's a really remarkable woman in that particular regard. I know we are sort of diverting a little bit, but I thought it was an interesting story to tell. So I admire Maryclaire King, more than one reason why I do so. A lot of people admire her too. All right, let's talk about the etiology of BRCA1 and BRCA2 genetics. Depending on the allele, okay, whether it's a truncation mutation, point mutation, and so on, race has a lot to do with it. In fact, we don't know why. The geographic location has something to do with it. We don't know why. So the epidemiology is really still very poorly developed in this particular regard. But obviously, the susceptibility is so dependent on the type of allele that you inherit. Long and short of it is that the lifetime risk of a BRCA1 individual, let's say, could be up to 80% in terms of developing breast cancer. All right, this is about 10-fold increase compared to the normal population. Unfortunately, 1 in 10 women over their lifetime would develop breast cancer, all right? But these individuals, for instance, if you have BRCA1 mutation, you tend to develop the more aggressive form of triple negative or breast cancer and so on. The etiology is really not well understood in that particular regard. In terms of ovarian cancer risk, it's up to 60%. It's about 24, okay, increase compared to the normal population in this particular regard. Now, how big is this problem? About 250,000 new cases of metastatic breast cancer diagnosed in the United States alone, only in the US, with about 50,000 deaths. Very grim. You would think that we should do better in terms of prolonging life and so on. We're doing better, but we have a long way to go in terms of prolonging people's life. Okay, ovarian cancer, about 45,000 new cases per year with about 16,000 deaths. Really, really dreadful and grim in this particular regard. Makes a strong argument that we need to understand the basic biology and try to translate some of the findings that we make. All right. Now, of course, BRCA1 and BRCA2 mutations are not just for breast or ovarian cancer. They're also very much relevant for understanding the pathogenesis of prostate. So this is not just a woman's health problem. It's a man's health issue as well, BRCA mutation. Okay, pancreatic cancer, okay, brain and even pediatric cancer such as neuroblastoma, believe it or not, okay, have been linked to mutations, okay, in these tumor suppressor genes. All right. Now, why do we come in? What has that got to do with us? Because BRCA1 and BRCA2, as I will tell you, and Lumia probably teaches this stuff, okay, locally and so on, and his colleagues probably teach this stuff, is that BRCA1 and BRCA2 are quite relevant, okay, for understanding the mechanism of DNA double-strand break repair. So this is a grim thing to study in a way, but scientifically it's a gold mine for us, all right. So we're trying to make some contribution in that particular regard. All right, I just want to point out that even though if you look at the number of frequency of mutation BRCA1 and BRCA2, they probably found in about 25% of hereditary, okay, type of breast ovarian cancer. They also account for maybe 5 or 6% of sporadic cancer, okay, in the breast and also in the ovary. However, tumors that don't have obvious mutations in these tumor suppressor genes, they often exhibit the same phenotype, like they're deficient or defective in DNA double-stranded break repair. The reason being the BRCA gene silence. You might have one good allele, even 2 good alleles, okay, or BRCA1, BRCA2, but nonetheless, epigenetically they are silenced, okay, in that particular regard. Or you have a mutation in the genes that normally encodes a protein that interacts with either BRCA1 or BRCA2, so that even though you don't have a mutation in BRCA1 or BRCA2, functionally speaking, you're BRCA deficient, okay, in that particular regard. One estimate, probably the higher limit, is that 1/3 of all tumor, 25% to 1/3 of the tumors have some deficiency, okay, in homologous repair, the topic of my presentation today. That may be the upper limit, okay. The actual number may be a little bit, okay, lower than that, but this is a big deal if you think about, okay, the prevalence of deficiency in homologous recombination-mediated repair in cancer biology. And of course, we're very glad that we have courageous people, okay, who come forward and not afraid to reveal, their family history and so on. And of course, we all know Angelina Jolie, right? She had her breast and her ovary removed. It's really grim, okay, as a prophylactic, measure. It's an effective prophylactic measure, but someone who have the important organs, removed like this in order not to get, cancer. So it's really grim if you think about that, all right. But she came out in the open and really make this a very high-profile thing, which is a good thing, to raise our awareness, right? I don't know how they estimate the 87% or 85%, but nonetheless, okay, that was what was reported for breast cancer and 50% chance ovarian cancer. So she had both her breast and her ovary removed in that particular regard. Let's talk about hardcore science now. First of all, I want to apologize. I'm going to show something very complicated. I'm trying to make it simple, all right? If I talk to the real expert who worked on this, my competitors and so on, they would say, you're missing out a lot of details, okay? But for us, okay, this suffices. In fact, this is quite complicated. Let me walk you through this particular scheme and focus on a couple of things that I would like you to make a mental note down, to sort of, because it will come back, not to haunt us, but it will come back as a theme on my presentation. Now, we studied DNA double-stranded break repair from the standpoint of understanding how a frank DNA break, for instance, if you express an endonuclease that recognizes a certain region of the chromosome, it cuts it, generates a clean break, all right? How that clean break is processed, okay, and repair. The reason why you want to do that is that the genetic system available, okay, will allow you to study the repair, the processing of the break in a more or less synchronized fashion. But double-stranded break repair obviously involving repairing ends that are dirty, like damage, all right, also repairing collapsed DNA replication fork, which is a different ballgame, all right? The enzymology is probably very similar, but the signaling would be very different when you're talking about repairing a frank chromosomal break or collapsed DNA replication fork. But it goes something like this in terms of the enzymology. The first step of the process is actually, okay, engagement of the ends by things like Ku, not shown here, and also a complex consisting of 53BP1, probably like 100 papers in Nature of Science have appeared on 53BP1, all right? We're beginning to understand more about what 53BP1 does. A nuclease is a complex of at least 4 other proteins, all right? Not shown here for simplicity. It blocks the DNA end from being accessed by the enzymes that function in homologous recombination, so as to channel the break into a different DNA repair pathway called a non-homologous DNA enjoining. Not shown here because I just want to focus on what we know about homologous repair. Now I want to draw your attention to BRCA1, BART1 being introduced the first time here. BART1 is a partner of BRCA1. I would be surprised if you find a situation where BRCA1 is not associated with BART1. They're probably obligatory, okay, partners. They form a heterodimer. They function together, all right? Inactivating one of the subunit akin to inactivating the whole complex in that particular regard. So BRCA1, BART1 have some unknown but well-documented function, okay, in the initial removal of the 53BP1 complex. How it does it? There are lots of speculation and so on going on, but nobody really knows. I think that's a fair thing to say, all right? There are people who argue that they know, but I don't think there's a lot of compelling evidence indicating how BRCA1, BART1 remove 53BP1. This is a complex that binds DNA, interacts with a lot of different things, and has a ubiquitin ligase activity. Whether the ubiquitin ligase activity somehow is involved in removal of 53BP1 complex, we don't know, but we're gonna address that, okay, in future. BRCA1, BART1 also functionally interact with some of the key components that involve in removal of 53BP1 and involve in so-called end resection. You take away one of the DNA strand, okay, to generate a long piece of single-stranded DNA. This is through the action of nucleases such as MR11, MRN complex, and also DNA2 and XO1. These are nucleases that act at different stages of the end resection reaction. The end result are 2 long single-stranded DNA tail. How long? In yeast cell, it will be kilobases, okay, in length. Mammalian cell probably shorter than that, maybe a few hundred bases or something along that line. The first factor that engages a single-stranded DNA is a protein called RPA. Bear in mind, RPA, because it's going to come back as I progress along a presentation of my data and results and conclusion and so on. RPA is a very abundant single-strand DNA binding protein, probably 10,000 copies per nucleus. It's incredibly high affinity for single-stranded DNA. We're talking about picomolar affinity, all right? So based on what we know about the property of RPA, okay, you would predict that that's the first factor that comes. Indeed, that's the case, okay, based on cell biological analysis. For homologous recombination to occur, RPA has to be replaced by a protein called Rad51. We were fortunate enough when I was an assistant professor, okay, in Texas in 1994. We were the first to document the activity on, in the Rad51 protein, showing its enzymatic activity, possibly linking its role, okay, in homologous recombination and DNA repair in that particular regard. Single author paper, as Lumi pointed out, very lucky. Happened to be at the right place at the right time. So Rad51 needs to replace RPA in order for repair to occur. This replacement is definitely dependent on another tumor suppressor, BRCA2. I'm going to focus on BRCA2 and tell you a little bit about, okay, what it does. BRCA2 is associated with a small protein called DSS1. Bear with me, I'll tell you a little bit about what DSS1 is and what it does. Genetic as well as cell biological evidence firmly implicated BRCA1, BART1, and other tumor suppressor called PALB2, acting in conjunction with BRCA2 to do the RPA to RAF51 handoff, or replacement of RPA, okay, with RAF51. How they do so, nobody knows, because of the difficulty involved in assembling the big complexes, okay, for biochemical studies and so on. I'm glad to report that we have been able to assemble these bigger complexes, including the pentameric complex consisting of all 5 proteins depicted here. I don't know exactly what it does yet, okay, but I'm hoping that within a year or so, we'll be able to find out. Once RAF51 is loaded onto the single-stranded DNA, it can conduct a genome-wide, okay, search for homolog. Most likely it's a homologous region in the sister chromatid, all right? Because of its proximity and also because of the signaling being favored in S phase cell when DNA replication is occurring. So once homology is found, RAF51, together with accessory factors, I'm going to show you some data today about BRCA1, BART1 being an accessory factor of RAF51 in homology search, as well as in the next step, creating a so-called strand invasion product. So RAF51 looks for homology, finds a homologous region, and catalyzes strand invasion, okay, to create this particular structure called a D-loop structure. So make a mental note of that. You're going to hear the term D-loop quite a number of times more as we go along. Okay, the strand invasion product then becomes an appropriate template to prime DNA synthesis. DNA synthesis serves the goal of replacing the information lost during the resection reaction and has a major impact on how the D-loop is resolved, okay? I'm not gonna go into the great mechanistic details about these different pathways and so on, because that necessitates another, okay, entire seminar, okay, in order to do that. Okay, I'm just gonna sum up. The D-loop can be resolved, okay, by different pathways and so on. Which DNA polymerase does the DNA synthesis? It turned out to be DNA polymerase delta. DNA polymerase epsilon has a minor role or no role to play. We don't know why that's the case, all right? So we have very good evidence now actually implicating BRCA1, BART1 in this step as well, right? It's still ongoing, okay? It's not close to publication and so on and so forth. Under some other circumstances, maybe we can discuss the work in that particular regard. All right. More recently, studies by a number of groups, okay, in the field, have unveiled another roles of the homologous recombination proteins, not in DNA repair, but in the active protection of DNA replication fork. It goes something like this, okay. Here you have a lesion or some sort of obstacle, could be a secondary structure, okay, present in the template strand. Now the obstacle is depicted as being on the leading strand. You can imagine if the obstacle is on the lagging strand. Similar things can happen, but the outcome may be different in terms of repair and processing. So here the lesion is located on the leading strand. Okay, there's number of one of several, okay, so-called DNA motor proteins that can recognize this type of structure and magically convert it into a DNA junction as depicted here. Has 4 arms, okay? One of the arms has a free DNA end. You want to protect that end, because if that end gets degraded, the whole fork would just collapse and all hell will break loose in that particular regard. It turned out that BRCA1 and BRCA2, presumably, okay, the associated proteins BART1 and PALB2 have something to do with this. So the current model posits that these tumor suppressors together as a unit, okay, load RAF51 onto the arm of the regress fork structure or the reverse fork structure, so the RAF51 can actually sit on the DNA and protect a barrier, okay, against the action of nucleus. Lumi is actually making quite significant progress in understanding, okay, how that occurs. You already have a couple of papers, okay, in that particular regard. We're more interested in whether BRCA1, BRCA2 acting as a single unit or their subcomplexes and so on in this particular regard. We're also interested in understanding if these proteins affect the activity of these so-called DNA motors. I bet they do, okay? There's no good genetic evidence that they do so yet, but I suspect that they do. Things don't normally occur like one step, like they normally talking to one another, okay, in biology. All right, so a lot of my presentation, and a lot of presentation by a lot of people who study homologous recombination, center on the ability of RAFD1 to form the so-called filament structure. It's a right-handed nuclear protein filament. RAFD1 forms this really beautiful structure on single-stranded DNA with a fixed pitch, about 90 to 100 angstrom, okay, with 6 protein monomers, okay, per helical turn, tied up about 18 bases of DNA, right? Studying this particular structure, it turned out to be a big topic, okay? Various people have been working on this for a number of years. We were lucky enough to be collaborating with a former colleague of mine. He's now the Dean of Life Sciences, okay, at Tsinghua University in China. They have incredible instrumentation. You have good instrumentation here too. They have 4 Titan Creols. You have 2, right? That's pretty good, right? But those guys are animals. Wow. They have just, wow. Yeah, the way they do stuff is amazing. They need room, they just dig a hole, okay, just create a basement to put the instrument there. In the US, all right, you can do, right? It would take like 20 years to get the permit to do it, right? But in China, they can do it, which is a good thing in a way, all right? So this is a cryo-EM structure of the RAFD1 filament. This is a reconstruction of the filament. We can spend like an hour talking about this. I'm not going to spend, a whole lot more time, just to show you that, okay, we can do reconstruction and so on. We have also been able to capture a catalytic intermediate when RAFD1 actually does the D-loop reaction. It brings in the homologous duplex, but the way we design the substrate, okay, the so-called invasion reaction is arrested because there's some non-homology in the substrate. Based on this, we can capture intermediate, really, really exciting. Unfortunately, the resolution is still very low, about 10 angstrom. We're trying very hard to get a better resolution and so on. I need to find my water. Okay, right here. Oh, forgive me. Okay, so this is ongoing. Of course, the longer-term goal is to see how BRCA1, BRCA2, and so on, okay, decorates the filament and the functional consequences of that. That would take ages, if ever, okay? It's much easier said than done. People who do structural biology will tell you that. All right, okay, that's that one. Okay, I told you some of this already, but let me just rehash a little bit. Formation of the RAFD1 single-stranded DNA filament is absolutely critical for subsequent steps to occur. That basically is a catalytic intermediate. Without a filament, no recombination, no repair, basically. Cell transformation, cancer, okay, you can extrapolate and all of that. So understanding how this filament is assembled is a big deal, okay? Long and short of it is that even though RAFD1 clearly binds single-stranded DNA, it does so with a very low affinity. We don't know why. That may create a rate limiting step for regulation to occur, perhaps, okay? But this is a very slow process. Because this is a slow process, it's prone to interference by RPA, which is very abundant and has very high affinity for single-stranded DNA, all right? So RPA gets in the way, so to speak. And you have to replace RPA, okay, in order to load RAFD1 to the single-stranded DNA. How does it do it? I'm gonna sum up many years of work by others and by us, and so on, in just 3 slides. Doesn't really do it justice, but that's the best we can do, given the circumstances. Okay, let me tell you a little bit about BRCA2, what it is, so on. You already know it's a breast tumor and ovarian tumor suppressor. Okay, it's an enormously large molecule, over 3,400 amino acid in length. It's enormous, huge, this thing. So it makes it very difficult to study it. We are probably only 1 out of 3 or 4 groups in the world that can reproducibly, okay, get following BRCA2 for mechanistic experiment. So what are the interesting features? There's a bunch of repeater sequences that are involved in RAF51 interaction. These interactions are very important for biological function. At the C-terminal region of BRCA2, there's another RAF51 interaction domain, okay, which we call CTRB, stands for C-terminal recombinase binding domain. We have a couple of very interesting stories about what it does, okay, but because of time, I'll be happy to tell you what the results are, okay, if you ask me later. Okay, so it has a bunch of other domains involved in interacting with the tumor suppressors and other partner proteins and so on. Let's forget about this protein called the MC1 because it's meiosis specific. BRCA2 also plays a role in meiotic recombination, but we're not talking about meiosis here, so I'm going to mention it and just move on. So the N-terminal region of BRCA2 is involved in interacting with the tumor suppressor PALB2. Okay, it has a DNA binding domain consisting of 3 of these so-called OB folds. These are protein motifs, okay, that are found in DNA binding proteins. So when you find them, most likely the protein is involved in binding DNA or some sort with some specificity and so on. Now, between OB1 and a helical domain, the crystal structure has been solved. That's why we know this is a helical domain. Defines a binding site for a very small protein called DSS1. If you wonder why is it called DSS1, it stands for deleted and split hand, split foot protein 1. It's a syndrome where individuals have developmental issues, their digits are fused. But DSS1 is not the protein mutated in the syndrome, but the name has stuck. Okay, go figure whether that's good or bad. Okay, but the name has stuck. Okay, so we refer to this protein as DSS1, all right? As large as BRCA2 is, okay, DSS1 is incredibly small. Just because it's small doesn't mean that it's not interesting. In fact, we find DSS1 to be incredibly interesting. Lumia has a very earth-shocking story about DSS1 outside of what I'm going to talk about. Okay, so DSS1 is small but very, very interesting in that particular regard. It's only 27 residues, highly acidic. How acidic? 27 out of 70 acidic. Either aspartate, okay, or glutamate. Go figure how acidic it is. Basically, it's half of it, okay, consists of acidic residue. The crystal structure of DSS1 bound to mouse BRCA2 has been solved. No reason to think that the human proteins don't, act the same way and so on. They're about 90% identical, all right? In this particular crystal structure, solved by Nikolai Plavatich's lab in 2002, published in Science, okay? The N-terminal region of DSS1, okay, in the C-terminal region, seem to embrace, okay, BRCA2, but there's a solvent-exposed loop, okay, that's highly acidic. We have shown by NMR, plus a lot of biochemical assays and so on, this is involved in interaction with RPA. It does so via DNA mimicry, acts as a DNA mimic. The end result is that it weakens the interaction of RPA on single-stranded DNA to facilitate the handoff. I'm really not doing very well on time. Am I? Okay. All right. Okay. So based on a lot of data, some of the data were published, okay, in a paper in 2015, okay, we can draw the conclusion that BRCA2 needs DSS1 or synergizes with DSS1 in handing off from RPA, okay, to RAD51 to form the RAD51 filament structure required for homologous repair. In this particular model, DSS1 is seen to contact the DNA binding OB fold, or RPA. I didn't have time to go into all the great details and so on, weakening the grip of RPA on single-stranded DNA, loosening up enough single-stranded DNA to prompt the nucleation of RAD51 on the single-stranded DNA. We know that once you have a few molecules of RAD51 on single-stranded DNA, it can nucleate the formation of the polymer, okay, of RAD51 without the help of other protein factors. So goes on very slowly, but filament growth, okay, occurs rather promptly. In so doing, it bumps off RPA. All right. So we think that the BRCA2 DSS1 complex plays such a role in this particular process. And of course, we're working very hard on finding out what BRCA1 does in this particular regard, what BART1 does, and what PALB2 does, because they together form a pentameric complex with BRCA2. That's ongoing. I'll be happy to tell you more if you wish. Oh, well, later on. All right. So that's BRCA2. Very fair to ask, what's the role of BRCA1 BART1 in homologous repair? If you look at my model, we place BRCA1 BART1 here, there, based on cellular evidence and so on. Long and short of it, we don't really know. This is after about 15,000 papers or so have appeared on BRCA1 BART1. 15,000 papers, okay, on these two proteins. Make a thought of that. This is really amazing. All right. We know very little. We are biochemists. We know of only one way to do, how to do it. You can call it not a very smart way, or you call it, this is a good way, but we know of only one way to find out how things work. Is to purify the proteins. Very simple. strategy. Worked out a way, purify enough of the protein, okay, and reconstitute, trying to put the reaction back together in that particular regard. All right, so the first author, okay, on this particular study, Wilson Zhao, I'm going to acknowledge him and so on later. He's absolutely incredible. I don't know how he does it. He works out all these, complicated procedure to make really difficult protein preparation and so on. He was also the first author on the study on BRCA2 that I just talked about. Just one person, he got it pretty much all worked out. All right, he's moving on now, looking at the E3 ligase activity of this particular complex. So he left no stone unturned. That's Wilson Zhao's approach to science. I don't care what people say, I'm going to look. Okay, I'm going to do the simple stuff to see if I find something that would be suggestive of the actual biological function. So he looked at DNA binding, all right? You might think he was crazy because there were like 3 or 4 papers on BRCA1, fragments of BRCA1, I should say, showing that BRCA1 has a DNA binding activity, but he wouldn't take that, okay, as an answer. So he looked very carefully to see if this particular complex would bind DNA, okay, and what types of DNA that it binds, okay? 2 major surprises, right? This has never been documented before until Wilson Zhao's work comes along. He found that not only BRCA1 BAT1 complex binds single-stranded DNA, it also binds double-stranded DNA. And if you look at different types of substrate, it binds the D loop with the highest affinity, okay? How high affinity? Really high, a few nanomolar in this particular regard. That's the apparent KD. We haven't done very rigorous, okay, biophysical characterization yet. In fact, if you look at single-stranded DNA, it's bound the least in that particular regard. That's surprise number 1. Surprise number 2, even though BRCA1 has been shown to bind DNA, this is a standard Southwestern analysis. You transfer the protein, you renature, and then probe the blood, okay, with a radiolabeled DNA substrate. If anything, BAT1 binds DNA, appears to have a higher affinity for DNA than BRCA1. We know that that's the truth because we have isolated the DNA binding domain from both proteins and done a very careful comparison. So BAT1 actually harbors the major DNA binding activity of this particular complex. All right, okay. Now this is somewhat of a surprise that it didn't actually bind single-stranded DNA with such high affinity. If anything, it has much higher affinity for the D-loop structure. And based on this, Wilson just had a very simple hypothesis that perhaps somehow is stabilizing the D-loop substrate or enabling RAFD1 to make the D-loop intermediate with high affinity and so on. A simple hypothesis. Good thing is, as Lumiere and others know, we have spent a lot of time working out all the in vitro assays that will allow us to address that type of question. This is one of those assays. It's actually very simple. We just take an oligonucleotide and throw in a plasmid molecule that has homology to the nucleotide, oligo, that we use as a substrate. And if RAFD1, with the help of accessory factors, can find a homologous region to form the D-loop substrate, it should assemble this particular intermediate, that looks like the D-loop and runs in a different spot of the gel, all right? So this was either the first or second experiment Wilson Zhao did. You can see, the effect is really dramatic. We see about 20 to 30-fold stimulation of D-loop formation. This is very specific because if you take the BRCA2 DSA1 complex in this particular regard, it didn't enable RAFD1, to make the D-loop more efficiently. Because its role, the role of BRCA2 DSA1, is in loading a RAFD1. Once RAFD1 loaded on the DNA, it doesn't seem to influence the activity of the RAFD1 filament. This quantification of the data. All right, so at this time, my longtime collaborator, Eric Green, came in, to provide another way to validate the result and add some mechanistic insight, so to speak. Want to give a shout out to Justin Steinfeld. I'm very proud because Justin was an MD student, a molecular biophysics student at Yale. He took my intermediate, biochemistry class. He was the guy just like me, sitting in front asking me tough questions all the time. I didn't like him very much initially because he gave me such a hard time always and asked good questions though that I often didn't have answer to, but that's okay. All right, so he became a MD PhD student in Eric Green's lab, and he did most of, or if not all of the single molecule analysis. Long and short, because of time and so on. So Eric has worked out the chemistry to deposit hundreds of these long single-stranded DNA molecule on a glass slide, flow in buffer with ATP, with protein and so on, to look for binding a protein to the DNA to look for things that move on DNA, how they move, how fast they move, and when they move, can they bump things off the DNA, that sort of thing. Okay, very versatile in that particular regard. This experiment setup, okay, it's actually very simple. It goes something like this. Rad51 is dark, it's not labeled, so it forms a filament on a single-stranded DNA. BRCA1, BART1, even though it's colored in green, it's also dark, it's not labeled. So you don't actually see it, all right? So, and then we flow in BRCA1, BART1, wild-type mutant, what have you, controls, and what have you, so on and so forth. Different types of buffer condition. And then the DNA target is labeled. Okay, so the DNA target, when it's captured by the filament, it shows up as a red dot. And then you just count the number of red dots per filament and look at the resident time. I'm simplifying it, okay? It's actually a lot more involved than what I've described, but that's the idea, okay? So some representative data are shown here. As you can see, BRCA1, BART1 greatly stimulates the ability of the rad filament to capture the duplex DNA, all right? Up to maybe 5 or 6-fold under these conditions. This is very specific. I will tell you the control later on. Based on kinetic analysis, we sort of deduced that it doesn't really affect the on rate. It didn't affect the off rate, I'm sorry. The resident time doesn't seem to be change in any significant regard. Okay, but the on rate seems to be the initial capture of the duplex target in that particular regard. I'm not going to show you all the data, but we run a lot of control. Okay, BART1 doesn't affect yeast Rad51. BRCA1, BART1 doesn't interact with yeast Rad51, and it doesn't affect the activity of yeast Rad51 in both the D-loop reaction and in the so-called DNA curtain analysis. I'm not going to show you all the data, because they're negative controls, essentially. Surprise number 2. Okay, we want to look for specific protein-protein interaction and so on. Now, it was shown in a Cell paper about 25 years ago when Ralph Scully, he's a professor at Harvard, working with David Livingston's lab at Harvard at the time, claiming that BRCA1 interacts with Rad51. Very exciting. Paper has been cited like 3,000 times, all right? But all of a sudden, citation dropped because people thought, oh, BRCA1 was bringing down Rad51 indirectly because they interact with BRCA2. That's why you could bring it down in IP experiments and so on. But our analysis, showed that there's actually direct interaction. This is a far Western analysis. You renature the protein on the blot and probe it with Rad51, just like a standard blot and so on. We have a lot more than this. We have the interaction domain and everything now. This is one of the assays. So BRCA1 actually directly interacts with Rad51. So that's exciting, confirming what Ralph Scully reported 25 years ago. But the surprise is that BART1 also interacts with Rad51. If anything, has high affinity, under these circumstances for Rad51. Rad54 is included as a positive control, and BSA, I believe, is included as a negative control in this particular regard. All right, so good, they interact and so on. That creates an opportunity to isolate mutants, all right? We need mutants to even just convince ourselves that we're looking at something biologically significant. So Wilson is really fantastic. In the morning, we discuss isolating mutants. Like after lunch, he walked to my office. I know what mutants to make. I said, really? Tell me. Okay, so he did all this alignment, and immediately you found a very highly conserved region that by protein domain mapping, he knows contains the Rad51 interaction domain in BART1, all right? Especially this phenylalanine residue that is found in a lot of Rad51 interaction, motif. So he was really excited about finding a highly conserved phenylalanine residue. So he went ahead and mutated the phenylalanine residue. He mutated 2 other residues as well. He called this particular allele AAE, designating the change to F to A, and then D to A, and so on, like that. He also deleted the whole, interaction region, that harbors the Rad51 interaction domain. And he's a very enterprising guy. He looked at cancer, mutation databases and so on. He told me, I'm going to make this one mutant, cancer-associated mutant. The reason being, this mutation has been found 4 independent cases of colorectal and uterine cancer. The people are not related. I mean, these are isolated, unrelated individuals, but they had the same mutation. We're going to call this, the K140M mutation, and that's your cancer-associated mutation. We did some cell-based assay to show that BART1, lo and behold, can bring down Rad51 robustly especially after treatment of cells, okay, with a DNA damaging agent such as mitomycin C. The AAE mutant expressed and examined under similar circumstances failed to bring down enhanced level of RAF51. So providing some sort of biological validation, this is significant. Now you will notice that BART1 becomes phosphorylated upon treatment of cells with mitomycin C. We don't know what the phosphorylation is for. It could be for RAF51 interaction, could be for something else, but we don't know that. We might address that. We might come around to address that. Now, it's time to examine the mutants that were isolated biochemically. There's a lot of stuff going on here, so I color-coded it, okay, for you, for easy viewing. This is a deletion mutant. We've done very little with it because of obvious caveat. If we have point mutants, we don't tend to work with truncation mutants. We did a limited amount with the truncation mutant, okay, in the D-loop assay. The AA mutant, okay, shown here, okay, and the so-called cancer-associated mutant, K140N mutant, okay, it's also similarly examined in the D-loop reaction. Let's look at the tabulated data. That's the wild-type level of stimulation in terms of D-loop formation. The internal deletion, the green, okay, line, you can see that's almost, okay, back all the way down, okay, the background level. The AA mutant is similarly impaired, although there's some residual, okay, activity. But even the K140N, the cancer-associated mutant, shows very, okay, significant deficiency in this particular regard, all right? Now, of course, we've done all the control, showing that none of these mutants affect DNA binding. That would be the obvious, caveat, okay, to eliminate. So we are pretty confident, okay? These are clean mutants, okay, that we could further work with. Okay, we've done a lot more with the AA mutant because it showed a very significant defect in vitro. We've done DNA current analysis also. I'm not going to show the data. It's similarly defective in that particular regard. Here we use two different types of biological assay. This is measuring, okay, intrachromatid, okay, recombination, okay, between two cryptic alleles, okay, of GFP. Recombination frequency is jacked up because of the introduction of a DNA double-stranded break, okay, through the introduction of the restriction endonuclease, ISC1, all right? So a bunch of controls, okay. So this would be a complemented vector alone. So this would be your baseline. This is complemented. These are all stable lines now, complemented with wild-type BART1, okay, elevates back up to near wild-type level. Good to see, all right? Expression of the AA mutant clearly has a deficiency in this particular regard. We know that the mutant protein is expressed. I'm not showing you the data at the same level. Goes into the nucleus without any problem. All right. So trivial explanation is eliminated. We've looked at the biological effect on recombination using a different system. This is gene targeting now. It's similar, okay, in terms of concept, but it's actually very different mechanistically. So you use, guide RNA, you CRISPR, and so on. You generate a site-specific double-stranded break, and then entire, okay, chromosomal recombination. This is actually a plasmid that you introduce in the cell. It's not really a chromosome, okay? Results in recombination, and then, readout. You just sort for fluorescent cell in that particular regard. Similar data, okay, complemented with empty vector. Again, these are stable lines, complemented with wild-type BART1, complemented with the mutant BART1. I don't have a whole lot more data, by the way, okay, in case you wonder. Now, there is certainly a deficiency, but only 2 to 3-fold. But I want to remind you, this is a single break. You repair a single break in euchromatin because the reported cell line was constructed to select those that give you a very high efficiency of recombination. Most likely the reporter was inserted in euchromatin. All right, so there's a single break repair and so on. Imperative for us to look at more sort of physiologically relevant breaks. For instance, induction of breaks during DNA replication by treatment of cells with mitomycin C. So now you have multiple breaks. Many of those breaks probably occur during DNA replication. Again, you're looking at stable cell line expressing a bunch of control up here. No need to walk you through them. It's just every control that you could, okay, imagine up here. Okay, the brown line is knockdown of BART1. Okay, the blue one is complemented of the knockdown cells with the AA mutant. Again, the AA mutant is expressed just fine, goes to the nucleus and so on. And you can see immediately that there's a lot of sensitivity to mitomycin C. So we think this is a better readout if you want to look at defect, okay, in certain mutants and what have you. And of course, you cannot publish a paper these days without looking at olaparib, okay, because the PARP inhibitors, olaparib is one of the inhibitors of an enzyme called PARP1. It's made its way into clinic, so everybody will be asking you to look at PARP inhibitor sensitivity, similar sort of a relationship in this particular regard, all right? Legitimate to ask me, okay, I know you're thinking, that's BRCA1, what about BRCA1? Is BRCA1 also involved in this particular regard? We have done some truncation mutant allele about BRCA1, assemble it together with BRCA1. Those truncation mutants don't work in all the assays that I described to you. So based on that, we suggest that interaction of BRCA1 with RAD51 is also important, okay, for the biological activity of BRCA1, BRCA1. All right, so this is my last slide, all right? So we found that BRCA1 interact with RAD51. We found that both proteins are involved in interacting with different types of DNA structure, including the D-loop structure, all right? In the literature, BRCA1, BRCA1 has been implicated in DNA damage signaling, somehow feedback, okay, provide a feedback signal to the ATM and ATR-dependent checkpoint, and so on. We're not working on this, okay? We're not expert in this, but we would be remiss not to mention it. BRCA1, BRCA1 somehow overcome the block imposed by 53BP1 and the shielding complex. I'm going to tell you just a little bit more before I finish up, and somehow influences the activity of the MR11 protein complex. This is one of the nucleases. We have evidence that it does physically interact and does, okay, functionally influence the activity of this particular complex. We're working very hard to figure out, okay, what BRCA1, BRCA1 does together with PALB2 and BRCA2 in loading RAD51 on the DNA. The red arrows pointing to the new findings, okay, that I've just described to you. Totally unexpected, because nobody could have predicted BRCA1, BRCA1 functions so late, okay, in recombination. We have evidence that it may be involved in DNA synthesis, okay, even later, okay, than the strand invasion step. It won't be surprising, okay, to me, anyhow. Okay, so I will finish up by telling you, okay, so where are we going next? Where we go from here? We have a long way to go. We probably know maybe 10% of what we need to know, okay, about these tumor suppressors. These are some of the things that we're doing. So hopefully with the new resources that we get, okay, when we relocate back to Texas, we'll be able to address some of them, or maybe all of them. Okay, the role of this in DNA, okay, and resection, how it overcomes the blocking effect of the shielding complex together with 53BP1, okay, looking at the pentameric, okay, complex, and RAD51 loading, and stabilization of the RAD51 fundament, so on, okay. I'm not going to walk you through every bullet point, but there's lots to do that would certainly keep me busy, okay, for a long time. If we're successful, okay, we may not be successful in any of these, but I suspect that we'll be able to provide more findings in this particular regard. Lots of people have contributed, okay, to the work that I talked about today. I'm not going to walk through, the whole slide. Just to give a special shout out to Wilson Zhao. Okay, he's just fantastic. Okay, he's actually coming to Texas as a tenure track faculty. He's got a very big, set of package and so on. So I'm really happy. He will have his own stuff going. We won't overlap, but we'll be helping each other and so on. So I thank you for your attention. I've gone over quite a bit. I will stop. Okay, perhaps take some questions that you might have. Thank you. Thanks very much.
Okay, thank you very much for disassembling the HR engine and putting it back together. And I'm opening the floor for the questions. Maybe I'll start with the first
Okay, warm up the audience.
Yeah, so that you can exercise a little bit before. I mean, you mentioned BRCA1, 2, all interacting with RAD51, and there is many more RAD51 associated protein reported, and they are all in protecting replication forks. So what's your imagination? Why do we need so many different factors affecting or stabilizing, loading, disassembling RAD51 activity?
Okay, so the simplest answer is it provides a reservoir of RAD51 because that nucleation event is still rare. Okay, it takes time for RPA to be replaced, even in the in vitro setting when we could artificially jack up the protein concentration and so on. I think whenever you have this, okay, DNA mimicry by DSS1, I suspect that PALB2 may also have a DNA mimicry type of attribute because it interacts with RPA, we map the domain, we just haven't made a mutant yet. So there are many different, several different ways, okay, of delivering the complex of RPA-coded DNA, and this reservoir, RAD51, okay, being located there. So when the single-stranded DNA is being free from RPA, you just have more of an opportunity to load RAD51. That's the simplest answer. The other scenario.
Okay.
All right, but the simplest, okay, proposal.
Yeah, thanks for wonderful talk. My question points to the basically 53BP1 protection of the ends, and you basically show that, or the question is, in case of mutants, in BRCA mutants, do you see a change in the pathway choice? Do you see change in resection?
Okay, change in resection. It's all centering upon the biology of 53BP1. So one of the major thing about 53BP1 is that without it, okay, homologous recombination goes up. Okay, so the premise is that by blocking in resection, it marks the break for repair by non-homologous end joining. Where the 53BP1 together with shielding complex are directly involved in end joining, possibly. Nobody has any data to that effect. We may address some of that when we have the system worked out. You know, we have everything assembled now. We just haven't done any mechanistic experiment. With shielding, I should mention we're collaborating with Steve Jackson at Cambridge on the shielding complex. So we're really cranking up to try to find out. Yes, pathway choices, I shouldn't even say primarily, but to a major extent is determined by whether there's resection. There's a well-known phenomenon of very little homologous recombination in G1 cells, okay, because there's very little resection in G1. The control is very complicated and not completely worked out.
So thank you very much. So you said that during homologous recombination in replication fork protection, the single-stranded DNA is being protected by Rad51 immediately. So how is it possible that it's not, I mean, the first factor which codes this single-stranded DNA is not RPA?
Okay, so you are talking about why doesn't Rad51 arrive on the scene before RPA does? Is that the question?
Yeah. I mean, why we don't have firstly RPA, which is
Why RPA first?
Yes.
Okay, by mass action. There's just abundance of RPA. Okay, Rad51 is quite abundant also, but if you do an in vitro assay, in cells it's more complicated, okay, because there are other things going on at the break. But if you just take a piece of single-stranded DNA and mix in both RPA and Rad51, and then find a way to pull out the single-stranded DNA to analyze what's on it, it's almost all RPA. The reason being, it just has such high affinity for the single-stranded DNA, picomolar, maybe 10 picomolar, 20 picomolar, or something like that. Rad51 probably high nanomolar. So there's 1,000-fold, okay, difference in affinity. Given the fact that it's very abundant, you know, I think the deduction is that that would be the protein, okay, that engages the single-stranded DNA first.
So maybe I misunderstood because I thought that you mentioned that in homologous recombination, in replication fork protection, we see only Rad51 and not RPA, no?
Okay, there must be RPA there. Conveniently missed out. This is all very recent. So, okay, for the leading strand at least, okay, when you do this type of fork regression, there's probably quite a bit of single-stranded DNA there. We don't know if all of that single-stranded DNA is stable, but clearly RPA would be there. You require BRCA2 to load Rad51, so the filament growth could stem from replacement of RPA by that BRCA complex, although it could be more complicated than that. Yeah, I missed it out. Yeah, that's a good point. Whenever the single-stranded DNA, more than likely there's RPA there. It's a great readout for single-stranded DNA. More reliable than anything else.
Maybe one more question from me. So you mentioned the later role of the BRCA1 and BARD1. So would it be possible, I mean, I guess you would need a particular separation of function mutants, but to to dissect this role also at the replication fork, so that the possible template switch and engagement with the second strand during replication.
So,
if
okay,
this is
so
something.
DNA binding mutant alleles that we're generating, probably defective in both homologous recombination and replication fork protection. That would be my prediction, okay? What you're asking is a lot more subtle, okay? Can we generate a separation or function mutation that predicates upon our ability to really understand the proteome, what it interacts with? That's why I bring in SMART cow and HLTF. I would not be surprised that BRCA1 BOT1 interacts with them, okay? Don't forget, it binds branch structure, okay, with high affinity. It could simply be stabilizing that, okay, junction point when it is first made. So if we do find an effect of BRCA1 BOT1, for instance, an enhancement of, let's say, SMART cow, all right, then we can map down the domain, generate specific mu, and see what happens.
Or perhaps you mentioned also the association with the polymerase delta with the DNA synthesis.
Yeah.
So again,
Yeah.
that interaction
Yeah.
may show the stabilize
Yes.
the exchange product.
Yes,
And,
yes.
and look at the replication.
There are opportunities for isolating a second function mutation for sure. You see, I'll just hold it. All right. For sure. That's a very valid question, but we may be 5 years away from getting there. Those mutants that are defective in rapid dual interaction, actually, I should say, someone walked up to me after the APCA meeting in London. You were there. I'm not going to tell you great detail because it's unpublished work, in saying that our AAE mutant, the BOT1 AAE mutant, has a replication fork phenotype. People move so fast. In a way, I'm glad, that the mutation that we described is actually useful for the field. In a way, it's also depressing that people move so fast. I'm not disclosing where the DNA binding domain is yet. Just to have a little bit of a, lead in that particular regard.
I would like to ask if you can compare the sensitivity of BRCA depletion to PARP versus to BRAD1.
Okay, so you're looking about the severity of the phenotype.
Yes.
When you get rid of BRCA1 and BOT1.
Yes.
Okay, which ones? They phenocopy. They
Phenocopy.
phenocopy.
So in that case, because there is a problem of BRCA reversal when you use PARP inhibitors. So in that case, can you use
I'm glad that
BRAD1?
you asked this question. So I'm part of a sort of a NCI, SPORE application. It's look at clinically relevant issues and so on. So some limited data out there, but sufficient to tell you that one of the major way of acquiring PARP resistance, or even cisplatin, resistant to cisplatin, is because the original truncation allele, because secondary mutations shift back into frame, but sometimes, the new allele stemming from those secondary mutations, they are far from full length, like missing like 1/3 of the protein. But the prevalent premise out there is that, well, that's sufficient to restore function, but we don't know that. We don't know that. We have been summoned to help out in that particular regard. It hasn't been on my radar screen because it's not very hypothesis-driven, but the clinical folks care a lot about it because they want to be able to find out whether those reversion events are really responsible for the acquired drug resistance in an effort to resensitize the tumor. So from that standpoint, it's actually important to know. But from our standpoint, we'll just be looking at a truncation mutant, and see what happens. But we're doing it. We will do some of that. Is that your question?
Yeah, I just meant that if the reversal happens of BRCA, in that case, can PARP sensitize BRAD1?
Would be attenuated, the sensitivity to PARP inhibitors. But people should really look at a whole panel. They should also look at the 53BP1 status. Not only that, silencing is costly, to do a whole panel of, epigenetic marks and so on, all right? Also look at shielding and so on. You cannot, I think, in a very sort of like critical way of looking at it, you shouldn't just look at reversion, or BRCA1 or BRCA2. You should also look at the status of shielding and so on, because it could be stemming from a combination of the reversion event or some epigenetic silencing, or overexpression of shielding, for all you know. But we know some of the targets, so I think in future, and we figure a way to fund it. You know, money is everything, unfortunately. If we can figure out a way to fund it, that's how I would do it.
Just very unrelated sort of question. The BRCA1 is also involved in meiosis. And here from your data, it seems like, you know, the homology search is really impaired in BRCA mutants.
Okay.
So how come that these people actually are not sterile? And how come that the meiotic recombination still works?
Okay, but a null allele wouldn't be even viable. Those must be hypomorphic allele.
Okay.
Okay, all
Okay.
right. I think the BRCA2 knockout, I think they probably knock out exon 27.
Yeah.
I don't think there's a null knockout. There's only one human cell line with a real null, okay, for BRCA2 called DLD1, I don't know. Okay, but has 1,000 other things going on in the cell line.
But the hereditary cancerogenic mutations, you think they do not affect the recombination in such a...
Nobody has looked at that carefully in meiosis. There's some old plant data from Marie, she passed away recently. Marie, the name will come back. Okay, in France,
Yeah.
they study meiosis, BRCA2. BRCA2 clearly has a meiotic phenotype. Dutrix, right?
Yes.
Yeah.
So if there are no more questions, please join me. Thank you one more time
Oh,
for your
thank you.
lecture and coming to
Thank you,
the
thank you. Thank you. Thank you.